DNA is one of the most important topics in biology. It codes for our traits and our genes, and it tells our cells what to do. And when it's time for our cells to divide, DNA is responsible for replicating itself and putting our genetic material where it needs to be. So let's take a closer look at DNA and what happens during this process. DNA replication is carried out by substances called enzymes. Enzymes help catalyze or speed up biochemical reactions in our bodies by performing very specific jobs. In this video, I'll go over all the enzymes that have a role in DNA replication and how they help speed up the process. Because DNA is double-stranded, helicase is responsible for splitting DNA so that each strand is on its own and the rest of the enzymes can perform replication on it. Helicase is also known as the splitting or unzipping enzyme, and once its job has been done, the DNA replication process can begin. Single-stranded binding proteins, or SSPs, have a very simple job. They're responsible for binding to split DNA strands and preventing them from coming back together during the DNA replication process. Next on the list is topoisomerase, with a bigger name and an even bigger job. Topoisomerase is responsible for preventing supercoiling. Supercoiling occurs when DNA overwinds, making it difficult for other enzymes to perform DNA replication. Now it's time for the new DNA construction to begin. This is DNA primase. Primase is responsible for making substances called primers, which serve as start flags for other enzymes and indicate where construction should begin. This is DNA polymerase. Polymerase is responsible for constructing DNA bases to help replicate and make new DNA. This is probably the most important enzyme in the entire process. Polymerase also has an ability to proofread its own work to avoid making mistakes in new DNA. Once new DNA has been constructed by DNA polymerase, you'll notice you have two different strands, one that's full and complete, and one that's split up into fragments. This is because DNA is anti-parallel, meaning that each separate strand runs in an opposite direction. Because of this, in the DNA replication process, we have two different strands. The leading strand, which runs from 5' to 3', and the lagging strand, which runs from 3' to 5'. These fragments that I previously mentioned on the lagging strand are known as Okazaki fragments. Okazaki fragments is DNA built in pieces because DNA polymerase only has the ability to build in one direction. This brings us to our last enzyme for this video. This is DNA ligase, which is responsible for gluing together Okazaki fragments on the lagging strand to make a full and complete strand like the one on the leading strand. Once ligase has glued together the Okazaki fragments, this marks the end of the DNA replication process. Before we go, it's important to note that this form of DNA replication is known as semi-conservative. Semi-conservative replication occurs when one DNA strand creates two new strands. Each one of these two new strands carries an original strand from the DNA, but they also each carry their brand new replicated strand. I hope this video was helpful and you enjoyed it. If you need more additional help, please feel free to check out the other videos on this channel. See you next time.